Everybody, so it's a big GI day today. We had internal medicine grand rounds this morning with Kim Brown, who did a fantastic job on portal hypertension. So I'm going to talk a little bit about some of my work and some ongoing research initiatives in the department at UofL that can impact trainees. And gosh, this is sensitive. Wow. And so as one of the jobs I've had as a vice chair for research is, and I appreciate seeing Jason Bell here, we worked on this together, was to create a strategic plan for research for the department. It's up here, and just so that you guys can see it and know that we have a strategic plan and vision for research in the Department of Medicine. And we want to have a research-based culture. We're going to track our metrics and try to improve them. We want to try to align university research and center goals, which are tied to specific service lines that we're not going to get into today. We want to have a broad portfolio of federal funding. And for the trainees, we want to provide research, education, and training. And we want to try to develop new researchers from within. And you can see me and Fred here from KET about a month ago. And so Fred's an example of developing new investigators from within. He's a postdoc in my lab and is gave great research grand rounds last week. So he's a success story, but we want to do this with clinicians. Man, Jason, this got like a keyboard sticking or something. Jason Puckett, he's gone. Guess I'll try a different arrow or mouse. I've never seen a PowerPoint so sensitive. So just so you know, some research metrics and just overview. So this is UofL. This was made a couple months ago. So these are UofL's physicians that are on federal grants, as particularly with leadership positions and roles. And so under division, these are in medicine, and the all other is other departments. And so you can see the list is not very big. We have a lot of federal grants at UofL, but most of those are to PhD investigators. In the Department of Medicine, you can see there's Klein, Castor, McLean, me, Abel, Dryden, Segal, Bowley, Chesney, Tenang, and Doster are on grants. So it's not a lot. And so we want to grow this list and have more. Yes.
And this is throughout the whole, just in medicine, or the whole, you had surgery and pediatrics?
Yeah, so the all other, so Jai-Ping Wong, Kyle Brothers, Sarah Watson.
But not like the whole universe, like the main campus or other campuses?
Yeah, these are all MDs.
Yeah, okay.
There's not many. These are not all the grants. These are all the grants from MDs.
MDs, gotcha.
Or DOs. It's not counting dentists and other types of healthcare providers. These are UofL physicians. And I don't think this hasn't been updated in a couple months. So I think there may be somebody from your division that's maybe on.
Well, we had the Traeger Institute Off-Line Clinic, but that's like their PhD social workers. They're like the PIs on the grant. Yeah.
Okay. So we want to develop more physician scientists from within. And so we have this whole trainee to faculty transition that Craig McLean has kind of come up with, leveraging some unique resources that we have at UofL. And so you can see, for residents to fellows to junior faculty, kind of progressing down this road on people's research career. And then we have unique institutional resources at each step of the way to get you there. And so for residents, they're eligible to apply for Kim's CREATE program. People that do, under Tom Miller, there was a what Kim calls earn while you learn, sign on bonus, so to speak, if you sign to join faculty, a stipend. Whether Jason Smith's going to continue that. I'm not sure. And if, what do we do if it's for a researcher? Will we still give any bonus, or is it prorated? I think it remains to be seen. But their point is that there are resources. Fellows are able to apply for CREATE, something called IDEA CTRD or LCTRC. They can apply for pilots, and as junior faculty pilots and developmental awards, and we've got resources called co-grades. There's also NIH repayment programs for MDs that are interested in research career, where they'll pay off your med school. They paid off mine, about 95%. I paid off. If I'd known that, I would have lived a better lifestyle as a medical student up through junior faculty. So anyway, I would like to get some of the medicine residents and medicines fellows, more engaged with these mechanisms in order to develop their research interest and develop them with clinician scientists so that we can retain them and grow from within. That's part of strategic plan. So here's some disclosures from me well, I guess it's an old picture from college up there. Not sure why I didn't take that one off. But don't really have any conflicts necessarily related to this part of the talk. It's about some of our environmental health research. There's a famous quote from someone named Rachel Carson: "As the tide of chemicals born of industrial age has risen to engulf our environment." This was from the 1960s. Drastic change has come about in the nature of the most serious health problems. We have had a change, with more obesity, more diabetes, over the last 20 or 30 years. And Rob Sargis, who gave medicine grand rounds earlier in the spring, made this figure. It's just looking at, this is totally correlative, doesn't prove any causality, but if you look at the chemical production, industrial chemicals by what are these millions of pounds or something over time, and diabetes prevalence over time, they're the exact same line, suggesting there could be an association, but not proven. Case presentation. This is one of my former patients, Paul Coder. Paul, that's his picture there, and I'm allowed to talk about him. Because we were in this article together, he agreed to be in this article and actually wrote a letter for the Department of Defense TERP toxic exposure grant that Dr. Watson and I have. So Paul was a Marine veteran. There he is in his home in Kentucky under a Marine flag. He was one of my clinic patients with fatty liver and cirrhosis, and he was at U.S. Marine Corps Base Camp Lejeune in the 1970s for a couple of years. You all may have seen ads, within the last couple of years. And were you ever at Camp Lejeune? Call this number for the tough lawyers, to take your case. Because there were toxic chemicals in the drinking water. They were solvents, some of the same types of chemicals that Sanjay Shrivastava and others study in the Superfund research program. Paul's brother Paul has two brothers that are also my patients. One brother died when I was on service at Jewish. He presented late with hepatocellular carcinoma. That brother was also exposed to toxic chemicals. He was a vinyl chloride worker at Louisville's Rubbertown complex. So anyway, Paul wanted to know, could any of these exposures to his brother, he worked at the chemical plant, or to himself when he was drinking contaminated drinking water with the volatile organic compounds in the drinking water at Camp Lejeune. So here's some of our objectives. Talk about occupational and environmental hepatology or liver diseases, and just have people recognize that pollution can impact liver health. This is not a new concept, and so here's some things over time. You may recall from medical school association between something called aflatoxin and liver cancer. It was first recognized in turkeys in England. It was called turkey X disease. Aflatoxin comes from rotten food, fungus-infected food. So there's some corn with a aflatoxin producing mold on it. There was something called Epping jaundice that happened in the 1960s, where a guy was delivering flour in a van in England and also delivering some kind of toxic chemical. The toxic chemical container dumped over and spilled on the flour. I don't know if anybody knew it or not. So they ended up taking the flour to bakery and baking a lot of stuff. And people that ate bread from the bakery turned yellow. And they called that Epping jaundice. It was in Epping, England. And then here in Louisville in the 1970s, they first made association between vinyl chloride exposures and hepatic hemangiosarcomas, which was really a sentinel event in occupational health. These patients still present from time to time. Here's a patient that I took care of with a giant liver mass, and this is a angiosarcoma of the liver and a chemical worker. There's about 30 of these cases, I think, per year in the country or worldwide. It's incredibly rare cancer. We've had almost 30 from this one chemical plant in Louisville, Kentucky, over time. There were other events in Taiwan and in Spain. The one in Spain was especially interesting. Somebody, I guess, realized that olive oil is a more expensive cooking oil, so it's priced higher. It's made out of primarily oleic acid. And so if you buy olive oil, it costs more than canola oil. And so they developed a chemical process to convert the canola oil into olive oil. And it got contaminated along the way with anilins. And they got something called toxic oil syndrome. They actually sold this stuff, chemically, modified canola oil turned into olive oil. And a lot of people got what was called toxic oil syndrome. And it was kind of a sclerosing type of disease. But it did feature some cholestatic liver disease. It was kind of like a systemic sclerosis. But they did get liver fibrosis and cholestasis. And then in 2010 was when we recognized fatty liver in the chemical workers in Rubber Town. And so this is kind of where I got involved as a trainee at UofL in the 2000s. So we had a very large biorepository, which we still have, from chemical workers that was initiated when the first cases of angiosarcoma were identified. This event in Rubber Town really kind of formed the basis for the Superfund research program, along with Aruni and Sanjay and Tim and others, Dan's research on VOCs, metals, and cardiovascular disease. They have to be tied to, that program has to be tied to a pollution Superfund site. And that site is called Lee's Lane Landfill, which was basically the toxic dump where the chemical plants in West Louisville dumped their chemicals. And so polyvinyl chloride production began in World War II by the Department of War. So I think Trump wants to change defense to the Department of War again. It used to be Department of War. And the Department of War authorized production of synthetic rubber in Louisville, Kentucky for the war effort, because Japan cut off the shipping lanes and we couldn't make tires for military vehicles because we couldn't get natural rubber. So we had to make synthetic rubber, and they put it here because it was inland, so it should be protected from air raids compared to a coastal location. And we were on the Ohio River, so easy ways to ship. And the large complex was operated initially by B.F. Goodrich and JAMA. 4 of the polyvinyl chloride or PVC workers were reported to have liver cancer. And a large health surveillance project was initiated by Carlo Tamburo, who's pictures up on the upper right there, with NCI support and support of the company. That research was fundamental in reducing the workplace exposure limits, and something called vinyl chloride standard was developed to do that. That was 1 part per million time weighted average exposure. These guys were getting thousands of parts per million before that. Now, Dr. Tamburo saved a lot of materials, included old liver biopsy specimens. The workers were identified through epidemiological approaches. The ones that had the highest risk to get the cancer, they were something called polycleaners. That was their job. They would climb in the tanks and clean them out and breathe in the gases. They weren't given respirators at the time. And so you can imagine doing cancer surveillance in the 70s was much different than today. Today, we would do CAT scans or ultrasounds or MRIs. They were looking at labs, and if they were abnormal, sometimes they would do a random liver biopsy, just hoping they might hit the cancer if the person had it. And so they actually biopsied a bunch of them. And that was kept in the biorepository. There were 2 boxes of old biopsies. Yeah, Christine.
I remember Dr. Timber. Did he die? Did he die of liver?
I think it was pancreatic.
Okay, no.
Here's what a picture of this tumor looks like. It's a rare tumor of the liver sinusoidal endothelial cell. So this is a cancer of endothelial cells for the cardiology researchers in the room or on the call. So you can imagine this cancer is trying to make little blood vessels, look like little blood vessels. That's a well differentiated one. So anyway, I looked at that box and I saw stuff like this. So while the cancer is what got everybody's attention, what was actually the most common was fatty liver in these workers, and they should have been protected by healthy worker effect. They weren't obese. They were relatively young. They at least claimed not to drink alcohol. And these are what some of their biopsies look like. This is steatohepatitis. Could be alcoholic hepatitis, could be non-alcoholic steatohepatitis, or the MASH or MASLD as we call it now. And this one actually has fatty liver with cirrhosis, a ring of collagen. This is a cirrhotic nodule with little fat droplets. The fat droplets are the white Swiss cheese looking things. And so this was 25 workers. I did this as a fellow in GI. 84% of them had steatosis, 80% had steatohepatitis, and over half had fibrosis. And so this is the prevalence of the steatosis or fatty liver is what you would expect to see in a bariatric surgery population. The BMI of these guys was like 26 only, maybe 25 and a half. And the steatohepatitis prevalence and fibrosis prevalence, to my knowledge, even though it's a small cohort, this is the highest, among the highest rates of steatohepatitis and fibrosis ever reported. Bariatric surgery patients don't get this much steatohepatitis. They get this much steatosis, but they don't get this much steatohepatitis, and they sure don't get this much fibrosis.
When you say bariatric surgery patients, you mean post-surgery or pre-surgery?
Well, hopefully.
Okay.
Bariatric patients. And come to Kispin next week if you want to learn more about bariatrics. We have a great program with Steve McClave. And so we called this TASH, toxic and associated steatohepatitis. Here's a picture of the complex today, and there's neighborhoods there, and that's part of the Superfund research program and the community engagement course. Residents are obviously concerned about their exposures. So we called this TASH. It's not every day, that you get to discover a new disease, but I did as a fellow. And so it's important for the trainees to know that some of your best work can happen even when you're a trainee, and don't sell yourself short. And so we created basically a new disease, and I've been studying ever since. Didn't know, when you're the only person looking at something, you don't know if you're just seeing stuff because you want to see stuff, if it's not there. But over time, and I've had several grants and awards aimed at increasing awareness of this type of problem. And now we have many other investigators looking in this field, and we're all finding stuff. And so that's been rewarding. And in 2019, this entered a practice guideline. So, we've kind of gone from discovery to actually entering this concept into a practice guideline in about a decade. So that's been a very rewarding part of my career. And so we've used a reverse translational approach in the lab, and along with some of our trainees, to develop animal models for the chemical exposures that we're interested in. And this is Juliana Beyer, who was here under Craig's Cobre, now at Pittsburgh, and she developed an animal model of diet, low-fat diet or high-fat diet with or without vinyl chloride, which is the VC. The red here is liver fat. This is a stain, pathologic stain. These are liver tissues stained for fat droplets, and you can see the greatest amount of red is in the high-fat diet plus vinyl chloride. So interestingly, you see in the low-fat diet, the vinyl didn't do anything. There's no increase in the red droplets, but there was some at least addition, well, it's not even addition, it's synergy with the high fat diet. And so this was at a very low level. These mice were exposed to less than 1 ppm. So they were given the exposure levels that OSHA allows people to get exposed to. And we've been seeing this in many other studies. There's nutrition interactions with these types of exposures. And she's used this model, her and others, to look at different mechanisms for the on how the exposure can result in the liver damage. And the exact mechanisms aren't important, but I think what's important to you is just to know that we've been able to replicate the findings in humans in a highly controlled animal model, and we've used that model to discover disease mechanisms, which we can then bring back to humans. So this vinyl has been in the news. So I think you've all heard of East Palestine. There's a new grant that's going to come out on that. Juliana Bayer that I showed is going to be a part of that grant and studying liver disease in East Palestine residents. Here's the backside of Ted Smith from Envirome. We've been at Calvert City. The same thing that happened at Rubbertown 50 years ago is happening currently as we speak in Calvert City. There's angiosarcomas, but this time they're not just in plant workers, they're in community residents.
And is that Western Kentucky?
Yeah, it's in Western Kentucky. Calvert City, Kentucky. It's just outside of Paducah,
Yeah.
Marshall County. And so here's Ted at the city council. And so Ted's going to get a R21 to study this problem down there through the community engagement course. So this is problem is unbelievably still going on at both a local and a national level. And so here's a figure from this practice guideline for occupational liver diseases. Here you can see various kinds of liver diseases here, histologic features, and some of the chemicals that have been associated with them. But the point is there's all kinds of liver pathologies that are associated with environmental exposures. Here's our TAFLD, which was toxic and associated fatty liver disease. I don't know what we call it now. They didn't pick up on this there was a new when they renamed the hash to MASH. So I call it MAFLD, MASH. I don't know what to call it. It's just another poorly named disease. And and what we believe is happening is that the exposures, if they're exceptional exposures to exceptional chemicals like vinyl chloride, if we've got in clinic, we've got Pseudomonas and Staph aureus, right? They're the king of the pathogens. Vinyl's like the king of the toxicants. So if you got the king, that could, I think, cause these diseases in of itself at a high exposure. But most of the lower level exposures may not cause the liver disease, but they can be considered to be disease modifying factors, just like diet, exercise, that type of thing. And we see that reproducibly. So we've been at over years asking questions, so we started with environmental health. We still do some environment, but we want to extend that out into the environment. And so if high level exposures to really bad chemicals in a chemical plant can cause liver disease, what about chronic low level environmental pollution or food contaminant exposures that all of us in this room get? Can that be a disease modifying factor or cause liver disease as well? And so we've been doing epidemiology. And so I had to learn how to be an epidemiologist, and not a very good one, but nobody else in the field was doing it. And so we had to do it. And so I probably have 15 papers on environmental epidemiology now, trying to find, what are some of these chemicals? We got about another 10 that need to be written. And so we used NHANES, and so NHANES actually measured environmental pollution, and we've got several papers on that, and we were able to go back and look for associations between environmental exposures and an ALT surrogate biomarker for fatty liver, and this was in 2010, and they had 196 pollutants, and we were looking at adjusted odds ratios for NAFLD using a surrogate biomarker. And we found these chemicals called polychlorinated biphenyls. And there's two subtypes. I'm not going to get into that. Were dose dependently associated with increased adjusted odds ratios for having fatty liver. So, and there's no, the first quartile is the referent because all of us are exposed to these chemicals. They're entirely man-made. They've been banned for like 30 years now, but they're a type of forever chemical. And so they're in all inhaled subjects. And so so there's no unexposed reference range, only first quartile reference range. But you can see as you go up into the higher exposure quartiles, your odds for fatty liver goes way up. And taking the same approach as we've done for vinyl, and Rita Wong, also on medicine faculty back when she was my graduate student, she developed an animal model where we did a similar approach to what Juliana did, looking at various diets and PCB exposure. And here, just like the vinyl, if you with this particular PCB, nothing really happened on a control or low fat diet. But when you gave a high fat diet, the animals got fat. So this was we called it a diet dependent obesogen. So this chemical was an obesogen, but only if you were on a bad diet. If you're on a high diet, so it made obesogen a chemical that makes you fat. And so it made the mice fat and it made the mouse livers fat, but only if the mouse was on a bad diet. And so Van Rita, who's here, these are some of my former trainees. They worked for about a decade using this and other models to find the mechanisms by which these chemicals cause this in the mouse model. And the list just goes on and on and on and on. But the short answer is that these are non-reactive chemicals. That's why they're forever chemicals. They put them in electrical transformers because you could put 1,000,000 volts of electricity through them and nothing happens. They don't break down. So P450 is not going to do much when a 1,000,000 volts of electricity is not. And so they last forever. But these things, through various mechanisms, they change, they reprogram liver gene expression. And that altered liver gene expression can be permanent. And there's many ways listed that we found that it can do that. And so these are really nasty chemicals. And right now we've been working with Carrie Kling in biochemistry, who's looking at post-transcriptional RNA modifications. She calls it epitranscriptomics. And so, and along the way, she's made a very important discovery, I think, because she's discovered a new mechanism for regulation of cholesterol. So I care about polychlorinated biphenyls. I don't think a lot of people do, but I think we should all care about new mechanisms for LDL, regulation. And so she's got a new R01 starting that I'm working on her. I think Mike and some of his cores will be helping with that too, because it's incredibly complicated. And so over time, there was no list of bad chemicals for liver that we're exposed to in the environment. So an agency called ATSDR publishes a priority list of what they believe to be the worst chemicals, and it's a balance of the toxicity of the chemicals versus the abundance or chance that you'll find these chemicals at Superfund sites and polluted sites across the country. And many of these chemicals that you can see their substance priority list rank is what ATSDR calls it. It's a government agency. For example, number 1 is arsenic. They rank it as the worst chemical. And I've listed some that have been associated with, actually all of these have been associated with liver disease and fatty liver, and at least some studies. Some of these were human, some were animals, some were both. And so we've been looking increasingly at different types of chemicals. We've had a lot of interesting collaborators and people that I've interacted with, and a lot of interesting cohorts. And so the World Trade Center first responders, they get fatty liver. And it's a little bit unclear if, and so this has been published and it was seen, they got, it's actually really interesting on the first responder cohort, because there's a fireman cohort, there's a police cohort, and I think there's another cohort too, but the different major universities in New York City basically own the different cohorts. and they don't share or cooperate with each other. So the firefighter people don't work with the police people. So I've been working a little bit with Mount Sinai and Andrea Branch, who's or at least advising her. And she doesn't need my advice. She's a great investigator on the firemen. And what they found was that the firemen were enrolled in a lung cancer screening project. And so they got periodic high-res CT chest for lung cancer. And those CTs got some lung windows. You scan the lungs, you get a piece of the liver. And they went back and looked for hepatic steatosis by CT scan. And they found a high prevalence of it. Sounds like some of those responders probably drank because of the trauma that they'd been through, and so how much of it is due to chemicals or how much due to alcohol? Of course, if it was Dr. Fred, he'd probably say it's the chemicals plus the alcohol, but not all of them. And so a very interesting cohort. Someone actually saved some of the dust from the World Trade Center, and Andrea and her team and collaborators at Sinai, they actually rubbed the dust on the nose of mice, and the mouse would lick the dust off its nose and swallow it. And they did an RNA sequencing of the mouse liver and were able to show that this dust, whatever it was, 20-year-old dust, still caused transcriptional changes in the liver, even though over 20 years, probably some of the really bad chemicals had kind of evaporated off of that dust and lost into the atmosphere. But that did argue that that dust was biologically active in liver, even though it was 20 years old and given by ingestion. So you can get liver toxicity from ingestion. If it's vinyl chloride gas, you can get it from inhalation too. And so that first cohort I did had 25 people, and now the cohorts are getting bigger. When I made this slide, there was a cohort of 90,000 people looking at PM 2.5 in China. Now the cohorts are literally up to millions of people. So people published on fatty liver and UK Biobank and other very large cohorts, and they're finding things. This was one on particulate matter. If you could do this, measure particulate matter in the atmosphere by geospatial technologies, satellites can find this, and then they could overlay that. This is in China, for example, here with CAT scans from whatever this was, 90,000 residents, and looking for fatty liver on CT. And so they found associations, and there were things that modified this association, like obesity, just like the mouse. If you have a bad diet or diet-induced obesity and you got exposed to the PM, it was bad. If you drank alcohol, it was really, really bad. If you smoked, it was really, really bad. But if you had high-fat diet, it wasn't quite as bad. And this work's gotten increasingly better. Like for some of these like UK Biobank studies, they've actually gone back and looked at omics. They have omics available. I can't remember what omics, if it's transcriptomics or proteomics or metabolomics, probably all of it, but they've actually gone back and found the associations with certain molecules and deciphered pathways that might be driving them. So a couple of key concepts is the liver is really number one organ for toxic chemicals. Here's data from EPA, older data looking at various tissues. And these were non-cancerous endpoints from environmental chemicals. And so liver is number one. So 25% of chemicals tested in the study caused liver toxicity. There's Dr. Merchant, so he'd be pleased to know kidneys number two in terms of toxicity. Neuro toxicity next, blood, thyroid, heart's down here. Dr. O'Toole's not speaking up. And so liver and kidney are believed, liver more so, but liver and kidney are involved with detoxification and metabolism of many of these chemicals. You don't think of clinically about kidney being metabolically active in xenobiotic metabolism, but it is, and that's why we see kidney toxicity from Tylenol, from acetaminophen. We worry about the liver, but if you take care of these patients, they all have high creatinines. And it's because of metabolism of Tylenol. And sometimes when the liver or kidney metabolizes a toxic chemical, it turns it into a worse chemical first before it detoxifies it further. It activates it through phase 1, and that's believed to account for this difference. I talked about the substance priority list, so 9 of the top 10 pollutants on that list have been associated with fatty liver. Environmental justice, I'm not sure if that's allowed to be a thing anymore, but there's heterogeneity, I guess is probably the term we could use now. There's environmental heterogeneity in terms of exposure. So different populations are exposed to different chemicals in different amounts and may have different susceptibilities to those exposures. And I'll show you some data on that if we don't run out of time. And those differences in exposures can lead to a differential liver response to exposure. And so that may be determined by race, ethnicity, sex, diet dependence, which we talked about, windows of susceptibility. When did that exposure occur? Was it in adult life? Was it in utero? Was it as a baby? And those windows, they matter. And that's probably because of different packing of the chromatin and accessibility of the DNA to chemicals themselves or receptors or other molecules that are changed by the interactions. But it's clear at this point that pollution has been associated with FAASLD development, severity, and complications. A little less on complications, but there are data. And again, we can consider these pollution exposures as just a disease modifier. Couple terms that are important for the House staff to know about is endocrine disrupting chemicals. So the Endocrine Society is really big on this. These are chemicals that are associated with diabetes and thyroid disease. And so they can disrupt any aspect of hormone action. These have been known for 20, 30 years. I'll talk a little bit about exposomes. So there's a newer omics technology called exposomics, where through mass spec, you can actually measure hundreds, if not thousands, of chemicals in the blood. We're working on a project with Emory where we routinely measure about 700 chemicals in blood, pollutant chemicals in blood that are annotated with confirmed with authentic standards. And they're there. Some of these are in 100% of people. We're working on that. Obesogens, I talked about, they're chemicals that cause obesity. I got into this through this toxicant-associated steatohepatitis or TASH. And now there's something called metabolism disrupting chemicals or MDCs. That kind of integrates all of these things. So, it's metabolic syndrome. So fatty liver is just part of metabolic syndrome. So you got chemicals that cause diabetes, chemicals that cause fatty liver, chemicals that cause obesity. And it's really all just metabolic syndrome. And so these metabolism disrupting chemicals disrupt normal metabolism and can promote any of these things: obesity, type 2 diabetes, high cholesterol, fatty liver, hypertension. These things are in all of us, and I can say that they're in all of us from doing these exposome studies. Some things we detect, bad toxic chemicals we detect in 100% of participants in some of our studies. We get them from food, we get them from industry, we get them from personal care products. We actually get them from medications and medical devices. A few years ago, there was a GI medicine that we used called Azicol that was withdrawn from the market. It's a medicine for inflammatory bowel disease. And then it came back later as Azicol HD. And nobody knew why it went off the market and why it was rebranded as Azicol HD. I don't know what HD actually stands for, but it should have been called Azicol phthalate free. And so that initial Azicol had a lot of phthalates in it, and not just Azicol, but many other extended release medicines have toxic chemicals in them because it's basically plastics that give it the time release. And so in the plastics were these plasticizers called phthalates, and this was accidentally discovered at Harvard. And so there was a guy there that was into reproductive toxicology and very interested in sperm motility and sperm function, reproductive health function. And phthalates have something to do with that. And he was measuring phthalate levels in patients, research subjects, and one subject had the highest phthalate levels that they'd ever seen and couldn't make sense of it, brought him back in, thought it was a lab error, tested it again, it was even higher. So what's going on with you? Do you have health problems? Who's your doctor? Well, I've got ulcerative colitis. My doctor's Josh Korsnick, IBD guy. And as it turns out, this guy was taking Azicol and getting phthalates and Azicol. And they actually ended up making a crossover study where they gave patients this phthalate containing Azicol and could show abnormal sperm function or counts in these patients that kind of change when they cross them over or switch them over, kind of confirming some effect. And, it's long been debated in IBD, and I'm not an IBD person, whether or not IBD is associated with male infertility or female fertility for that matter. And I'm not saying it is or it isn't, but I'm just saying if it is, it could be potentially due to some of the toxic chemicals that we were inadvertently giving patients with our medications. People also got plasticizers through or get them. It's less of a problem now, but through IV tubing and bags. And you can now, if you just Google it, it's in the news periodically, how many microplastics you get from getting a liter of IV fluid. So that's been in the news lately. Every time we give them an infusion, we're giving them microplastics. Every time we give them fluids. Many mechanisms for obesity, so caused by pollution. So any mechanism for obesity you can think of, there's been a chemical shown to work through that mechanism. DDT is a concerning one. It's an old, you've probably heard of that. DDT, it's been banned. It's another forever chemical. And this investigator here, Michelle Lamerrill, she showed that, and this is scary stuff, that they had this cohort of grandmothers that was archived blood samples, and they followed up the grandchildren, and they could show that obesity in the grandchildren was associated with the DDT levels in the mother. And it was just the girls, so it was sex-specific effects. It didn't bother the boys, it was just the girls. So if you were an obese 25-year-old girl, it was more likely that your grandmother had high DDT levels. And so that's really scary because and she was able to show a similar thing in mice, which I'm not going to get down to. So these things are like passed through generations. And so this is really a scary concept because it suggests that some of the obesity and metabolic syndrome epidemic that we see today could be related to junk that our parents and grandparents and great-grandparents were exposed to that we can't control. But now we got to deal with it. And that's really scary. And I'll show you some liver-specific data for it. And again, the mechanisms are complex, but again, they tend to center ultimately around changing, reprogramming normal liver gene expression. It's kind of the common pathway for them. Now, this is the scariest one in liver. These are the PFAS forever chemicals. And this was done by the P30 Center at NIEHS P30 Center in Southern California for Lita Chatsey. So this was another multi-generational cohort. This was from Scandinavia. And so they drew blood on pregnant women, and then they followed up the kids. And they waited till the kids were adolescents. I can't remember exactly how old they were, probably 10 to 15-ish years old. And then they went back and they measured the forever chemical levels in the mom during pregnancy. And so the babies in utero were getting exposed to presumably whatever level that mom had in their blood. And then they went back to see, they were able to see which kids had abnormal liver enzymes and were at risk for pediatric fatty liver disease. And then they went back to look and see if there was any correlation between the fatty liver in the adolescents risk and mom's pregnancy forever chemicals levels. And so I try to be a toxicologist. Fred's a toxicologist. I try to be a toxicologist. And from my limited tox understanding, I would call this a perfect dose response curve. So the more higher the mom's forever chemical levels, the more likely the kids were going to get fatty liver. And they did metabolomics on these. And so these orange boxes are the forever chemical levels. And then these circles are metabolites. And so you can see that mom's forever chemical levels changed the blood metabolomics in the kids a decade later, just suggesting that there's a reprogramming of metabolism from in utero exposure that persisted. And this was highly controlled. They also measured the forever chemical levels in the kids and somehow took that into account in their statistical model so that you were confident that this wasn't the forever level, that forever chemical levels in the children, it was mom's forever chemical levels. And so this is incredibly scary again, suggesting that some of our health problems today were due to stuff our parents were exposed to, which we can't control. And let me just skip forward a little bit for sake of time here. I'll show you a little bit of some exposomics work we're doing. And so just some representative examples. So through mass spec, our collaborators at Emory can measure hundreds of pollutants in blood. We find them in our liver disease patients, and they do metabolomics as well. So those of you all that come to our clinics, clinicians, residents, will know we get the fiber scans a lot, and they were talked about in grand rounds today. Here's a fiber scan parameter that was discussed in medicine grand rounds, the liver stiffness measurement. And so you can see these chemicals here, PCB, diphenyl nitrosamine. These levels of these chemicals in blood end up being associated with the liver stiffness measurement and end up being associated with other metabolites in the blood too. And so we're finding that in multiple cohorts. Here's an example of a larger cohort, 325 subjects, and you always get junk you never heard of, that's floating around in bodies of our patients, probably ours too. There's something called iprodione in this study that came out. It's a fungicide. And what does it do? Well, if you look at the metabolomics of the iprodione, this is called a metabolome-wide association study versus levels in the blood of iprodione. There was 792 metabolites in blood that were significantly associated with the iprodione levels. And if you do a pathway analysis, you can see that iprodione was associated with changes in all of these pathways, some of which impact liver disease. So we've seen this for other chemicals as well. We're not just stuck on FAASLD. Fred talked last week about alcohol, and here's Tyler. I think he discussed Tyler's paper. He was one of our pre-doctoral students who did a fantastic job establishing models of alcohol. So environmental pollution doesn't cause alcohol-associated liver disease. Alcohol does, but environmental pollution may modify the alcohol-associated liver disease. Shift gears a little bit to talk about some of our work on military and veterans health. The case presentation was a veteran that was at Camp Lejeune that had cirrhosis due to NASH and had the brother that worked at the vinyl chloride plant that died of cancer, presumably from NASH. And so interested in veterans health. Camp Lejeune we discussed already, so Dr. Watson and I have a grant on Camp Lejeune exposures. He's doing animals, I'm doing humans. Within the last couple of years, there have been some congressionally directed medical research programs. They call them TERPs and MERPs. And so I've kind of some ways led a group at UofL in applying for these things. So we now have 6 awards on military and veterans environmental health. There's 4 active. You can see the investigators there that have these. We've got 2 additional notice of awards or notice of intent to awards. So 4 + 2 is 6. We've got 2 more that are in preparation. One from Dr. Dryden in Department of Medicine, which was very exciting, looking at active duty pilots, and another exciting one from Jai-Ping Wang in anesthesia, looking at pulmonary hypertension and environmental exposures. So a wide variety of grants. We've got looking at very many different military exposures, burn pits, some of the grants looking at burn pits, some are looking at things called dioxins, which are Agent Orange related chemicals, solvents, like the Camp Lejeune ones. The two pending ones are looking at the PFAS forever chemicals. These grants are highly collaborative, looking and involve multiple VA sites, multiple universities, military as well. So what are some of the future directions for my work? So my research has been supported for a very long grant called R35 which is expiring. And so I spent last year writing a lot of grants, and it looks like a lot of these are either recently funded or we expect them to get funded. So we have this one with MPI with Carrie Kling on this epigenetic regulation of cholesterol metabolism and liver disease and environmental exposures. Again, it's not the exposures that are important here. I think it's this novel mechanism for cholesterol regulation, and hopefully we'll find a druggable target on that and can do something really impactful. I talked about Calvert City, which is the new rubber town, and angiosarcoma. Ted Smith's getting an R21. I showed you a picture of Ted at the city council there on that. We've got a notice of award to be a partnering PI in a new DoD grant, which is looking at the treatment of veterans with high PFAS levels. And some of them are going to have very high PFAS levels. So the PFAS is a much bigger problem for the military than Agent Orange ever was, because unlike Agent Orange, which we dumped on Vietnam, we dumped PFAS on ourselves. And so it was in firefighting foam. So all the air bases get fires, fuel fires. They have PFAS containing firefighting foam that they sprayed, and it's a widespread contamination from that. And so we're developing a therapy, or at least testing a therapy to decrease PFAS levels in highly exposed veterans. We've got a pending VA merit to do longitudinal liver and metabolic effects of PFAS in veterans. And then we sent in a Nutrition and Metabolic Health Center, COBRA, several of you in the room helped with that, Brad Hills and MPI on that. And so that is a big center application that had a good score. And so we hope that might get funded. We don't know yet, but it's going to look at 5 areas of nutrition. And nutrition, again, I had throughout this talk because it interacts with the exposures as demonstrated by the data. And so we're going to look at nutrition therapy, intermediary metabolism, environmental health. We don't call it environmental health anymore. We call it food and water contaminants. For Kennedy, not Dr. Kennedy's sake. Looking at food intake, obesity, and Sherry Levinson, who gave a great talk at CTRB earlier this week, her group on eating disorders and vulnerable populations and equity, or whatever we're allowed to call them now. And so we have several research projects in the center and the Apple, and then cores to do some unique things related to nutrition. Dr. Merchant's in the room here, so his core is going to work with, or proposes to work with Melissa Smith and do some really neat new things that are kind of integrating the transcriptome and the proteome and looking at the steps in between, going from the mRNAs and going into the protein level, things that I don't quite frankly understand, but they sounded really cool. So appreciate that. So we'll have a lot of neat nutritional things and opportunities for house staff and junior faculties. It's more of a junior faculty development grant, but house officers can certainly participate in some of the projects. So what can you do if you're exposed so you can reduce your exposures? Organic diets were shown in one small study in children to reduce pesticide levels. You can eliminate some of these MDCs, metabolism disrupting chemicals. You can eliminate them by increasing their clearance. And so the number one is the strategy we're taking in our new Department of Defense grant for forever chemicals. We were looking at a bioacid binder. They also will bind the PFAS and make you poop them out. But Olestra will do that too. And some fermentable fibers have been shown to do that. And you can, there's limited data, but I think it's kind of cool, increase renal elimination. So maybe Dr. Merchant can help us with something like this, but SGLT2 inhibitors will increase urine output, and they'll apparently drag some of these toxic. So usually these toxic chemicals and metabolites either go through, urine or stool. So the stool ones you could trap in the bowel through something like cholestyramine. The renal ones, I guess you could increase, urine flow with SGLT2 and drag some along. And they're better than the good old fashioned way. So the good old fashioned way applied to women. So if you're a woman, you could have babies and you can give your chemicals to your babies either across the placenta or through breastfeeding. A lot of the chemicals come out in breast milk, and there's people that have made careers measuring toxic chemicals in mother's breast milk. And so it's a real thing. So there's many barriers to implementation, so we don't have an EPIC, a button that says measure forever chemical levels or any exposome, and there's no ICD code that's going to guarantee coverage for that. And then once it comes back, what are we going to do about it? Because they're not in guidelines. So we give talks at the meetings. There's in the NASH fatty liver practice guidelines that were published a year or two ago. They don't make any mention of it. So there's nothing in the practice guidelines. And so, we really need to take the next step to have clinical impact and overcome some of these barriers. And so we want to work toward that now. So kind of in conclusion, we have a strategic plan for research at UofL. Part of that's going to be increasingly looking at military and veterans health, and the EVPRI I think is interested in this as well. I met with him and Kevin Merton with Dr. Dryden about a week ago on this. Clinical and translational research is going to be very important, and we want to get house staff working with our investigators to use this unique career development roadmap that's available basically nowhere else. To get them to become physician scientists, because as I showed on an earlier slide, we don't have very many of us at UofL, MD, NIH funded investigators, and we want to increase that. And then I think I've showed along the way that pollution's bad for your liver, and certain barriers do exist. And I think this exposome concept, could become increasingly important. And the research has been a very rewarding part of my career in this area, as we strive to go, from occupational health to environmental health to bedside ultimately. And so this is from the liver meeting. This is our major liver meeting society that our speaker this morning is a real leader in. And so when I started going to this meetings and presenting these posters, they had them literally in the last aisle by the bathroom. And so no one ever came to my poster unless they had to go to the bathroom. And then they just accidentally, when they walked out of the bathroom, they were looking right at it. So I was always grateful when people had to go pee, because they'd come look at my poster, even if they didn't want to. So we were kind of like outcasts. But then they actually featured, some of our work on environmental toxins and liver disease at the best of the meeting. So we've come a long way, but we still got a ways to go.